Hello and welcome to Nighttime Science. Tonight we are stepping into a world that exists all around us yet often goes unnoticed. We will shrink our perspective down to explore a realm of delicate wings and intricate antennae, of compound eyes and jointed legs, of ancient lineages and remarkable adaptations. We are entering the world of insects and bugs, the most successful animals ever to walk, crawl, or fly upon this earth. There is no need to do anything now except to listen, to find a comfortable position, to allow your breathing to become slow and regular, and to let the gentle weight of relaxation settle upon you. As we begin our journey, imagine yourself becoming small enough to walk beneath a blade of grass as if it were a towering tree. The air around you hums softly with the distant drone of wings. The ground beneath reveals itself not as dirt, but as a landscape of particles and tunnels, of hidden chambers and pathways worn smooth by countless tiny feet, over countless generations. Tonight we will explore the quiet lives of these remarkable creatures. We will discover how they breathe without lungs, how they see the world through thousands of lenses, how they communicate in languages of chemistry and vibration. We will learn about their ancient history, their complex behaviors, and the essential roles they play in every ecosystem on Earth. Let your mind settle into curiosity and calm as we begin. The ancient origins of insects. The story of insects begins in a time so distant that the continents themselves were arranged differently upon the face of the earth. Long before the first dinosaur walked, before the first flower bloomed, before even the first trees reached toward the sky, insects had already begun their journey through evolutionary time. They are among the oldest groups of animals to live on land, and their history spans more than 400 million years of continuous existence. The earliest insects emerged during the Devonian period, roughly 400 million years ago, when life was just beginning to establish itself firmly on land. The world they entered was vastly different from our own. The continents were clustered together in configurations we would not recognize. The landscape was dominated by primitive plants, early ferns and horsetails, and the first forests were beginning to take shape. Into this world came the ancestors of all modern insects, small arthropods that had made the transition from water to land. These first insects were wingless creatures, simple in their body plan but already possessing the fundamental characteristics that would define their descendants. They had bodies divided into three segments: the head, thorax, and abdomen. They had 6 legs attached to the thorax. They had an exoskeleton made of chitin, a remarkable substance that provided both protection and structure. These features, established hundreds of millions of years ago, persist in every insect alive today, from the smallest parasitic wasp to the heaviest beetle. For millions of years, these early insects remained bound to the ground and to low vegetation. They crawled through the leaf litter of ancient forests, they burrowed in the soil, they climbed the stems of primitive plants. But then, approximately 360 million years ago, during the Carboniferous period, something extraordinary happened. Insects developed the ability to fly. They were the first animals ever to achieve powered flight, preceding birds by more than 100 million years and bats by more than 200 million years. This development of flight was a turning point not just for insects but for life on Earth. Suddenly, insects could escape predators by taking to the air. They could find new food sources in the canopy of forests. They could disperse across vast distances, colonizing new habitats and evolving into new forms. The evolution of flight opened up three-dimensional space as a realm for life, and insects were its first explorers. The Carboniferous period, lasting from 360 million to 300 million years ago, was a time when insects reached sizes that would seem impossible today. The fossil record from this era reveals dragonflies with wingspans reaching 70 centimeters, nearly the width of a large dinner plate. There were millipedes, though not technically insects, stretching more than 2 meters in length. Giant mayflies, enormous cockroaches, and other arthropods of striking proportions inhabited the swampy forests of this distant age. The reason for these enormous sizes has been a subject of scientific investigation for many years. The prevailing explanation relates to the composition of the atmosphere during the Carboniferous period. Oxygen levels in the air were significantly higher then than they are today, perhaps as high as 35% compared to the 21% we breathe now. This enriched oxygen environment had profound effects on insect physiology. Insects do not breathe through lungs as we do. Instead, they rely on a system of tubes called tracheae that carry oxygen directly to their tissues. This system works efficiently at small sizes but becomes less effective as body size increases because oxygen must travel farther through the tubes to reach internal tissues. However, when atmospheric oxygen is more abundant, it can diffuse more readily through these tubes, allowing the system to function effectively even in larger bodies. The high oxygen levels of the Carboniferous period thus permitted insects to grow to sizes that would be unsustainable in our current atmosphere. As the Carboniferous period gave way to the Permian, and as oxygen levels gradually declined, the era of giant insects came to an end. The massive dragonflies and enormous beetles disappeared from the fossil record. But insects themselves did not diminish in importance. They continued to diversify, to adapt, to evolve into the astonishing variety of forms we see today. The Permian period, from 300 million to 250 million years ago, saw the rise of many new insect groups. Beetles began to appear. True bugs, the order Hemiptera, emerged. The ancestors of modern flies and wasps began their evolutionary journeys. Each group developed its own specializations, its own ways of feeding, moving, and reproducing. Then came the great extinction event at the end of the Permian, the most severe extinction in Earth's history, when more than 90% of all species disappeared. The causes of this catastrophe are still debated, but its effects were devastating. Yet insects survived. They persisted through this crucible and emerged to inherit the recovering world. The Mesozoic era, the age of dinosaurs, was also a golden age for insect evolution. As flowering plants appeared and diversified during the Cretaceous period beginning about 140 million years ago, insects and plants began the intricate dance of coevolution that continues today. Bees evolved to pollinate flowers. Butterflies and moths developed specialized mouthparts to sip nectar. Plants in turn evolved colors, scents, and shapes to attract these insect visitors. This partnership between insects and flowering plants would eventually reshape the entire botanical landscape of Earth. Through the Mesozoic and into the Cenozoic, through the age of mammals and the rise of humans, insects continued their steady existence. They witnessed the asteroid impact that ended the reign of the dinosaurs 66 million years ago. They survived ice ages and warm periods, continental drift and volcanic eruptions. They adapted to deserts and rainforests, to mountains and caves, to every terrestrial habitat Earth has to offer. Today, insects represent the most diverse group of animals on our planet. Scientists have described approximately 1 million species of insects, but estimates suggest the true number may be several times higher, perhaps 5 million or even more. They account for more than half of all known living organisms. For every human on Earth, there are an estimated 1.4 billion insects at any given moment. Their combined biomass exceeds that of all humans and all livestock combined. This incredible success, this persistence across hundreds of millions of years, speaks to the fundamental soundness of the insect body plan. The division of the body into 3 segments, the 6 legs, the exoskeleton, the ability to fly. These features have proven remarkably adaptable. They have allowed insects to evolve into forms as varied as the armored rhinoceros beetle, the delicate lacewing, the streamlined water strider, and the social honeybee. Some insect lineages alive today are extraordinarily ancient. Silverfish, those small, silvery insects sometimes found in old books or damp bathrooms, belong to one of the most primitive groups of insects still in existence. Their order, Zygentoma, has remained relatively unchanged for more than 400 million years. To see a silverfish is to glimpse a body form that existed before the first dinosaurs, before the first trees in the earliest days of terrestrial life. Dragonflies, too, are ancient. Their order, Odonata, dates back approximately 320 million years to the Carboniferous period. While modern dragonflies are smaller than their prehistoric cousins, they retain the same basic body structure, the same incredible flying abilities, the same predatory lifestyle. When a dragonfly hovers over a summer pond, it performs maneuvers that its ancestors perfected hundreds of millions of years ago. Cockroaches represent another lineage of great antiquity. Fossils of cockroach-like insects have been found in rocks more than 350 million years old. These ancient roaches inhabited the coal swamps of the Carboniferous, living among giant ferns and primitive trees. The fact that cockroaches still exist today, largely unchanged in their basic form, demonstrates their success as a design for survival. Mayflies, despite their brief adult lives, belong to an order called Ephemeroptera that extends back more than 300 million years. The ancestors of modern mayflies flew above ancient waterways when the world was young, and their descendants continue the same patterns today, emerging from streams and rivers in mass flights that have occurred every summer for geological ages. As we contemplate these ancient lineages, we begin to understand that insects are not merely small creatures going about their lives in our gardens and forests. They are living links to Earth's deep past. They carry within their bodies, in their genes and their structures, the accumulated wisdom of hundreds of millions of years of evolution. They have solved problems we are only beginning to understand, problems of efficiency, of resource use, of survival in a changing world. The exoskeleton that protects a beetle today is built from the same chitin molecules that armored the first land-dwelling arthropods in the Devonian. The compound eyes through which a fly views the world use the same basic design as the eyes of Carboniferous dragonflies. The wings that lift a butterfly into the air operate on aerodynamic principles discovered by insects in the deep past and refined over countless generations. In quiet moments when we observe an insect going about its business, we are witnessing not just an individual organism but the current expression of an evolutionary journey spanning epochs. We are seeing the result of uncountable generations, of adaptations tested and retained, of innovations that worked and were passed down through the flow of time. Each insect is both utterly itself, living its particular life in this particular moment, and simultaneously an embodiment of deep history, a creature shaped by forces and events that unfolded across the vast canvas of geological time. The continents have moved, mountains have risen and worn away, oceans have advanced and retreated. The climate has warmed and cooled in great cycles. Through all of this, insects have persisted, have adapted, have thrived. Their success is not an accident. It is the result of fundamental advantages built into their body plan, advantages we will explore as our journey continues. But for now, simply rest in the knowledge that the insects we will discuss tonight are heirs to one of life's greatest success stories. They are the descendants of pioneers, of the first animals to fly, of creatures that have survived every challenge Earth has presented for 400 million years. Their story is one of resilience, adaptation, and quiet persistence, and it continues all around us in every season, in every habitat, at every moment of every day. The insect brain and nervous system. Within the tiny head of an insect lies a brain, small beyond our usual conception of what a brain might be, yet capable of remarkable feats of processing and control. The brain of a fruit fly, one of the most studied insects in science, contains approximately 140,000 neurons. A honeybee's brain holds about 1 million neurons. These numbers seem minuscule when compared to the roughly 86 billion neurons in a human brain. Yet these compact neural structures enable insects to navigate complex environments, learn from experience, communicate with others of their kind, and execute behaviors of surprising sophistication. The insect brain is divided into several distinct regions, each with specialized functions. The largest and most prominent structures are the optic lobes, which process visual information from the compound eyes. In insects that rely heavily on vision, such as dragonflies and bees, the optic lobes can account for a significant portion of the entire brain. These regions contain layers of neural tissue arranged in columns, creating a structure that processes motion, color, and pattern with remarkable efficiency. Above and between the optic lobes lies the protocerebrum, the most complex part of the insect brain. This region integrates information from multiple senses and plays a role in learning, memory, and higher-order behaviors. Within the protocerebrum are structures called mushroom bodies, which are particularly well developed in social insects like ants and bees. The mushroom bodies are essential for learning and memory formation. When a bee learns to associate a particular flower color with a nectar reward, the mushroom bodies encode and store this information, allowing the bee to make better foraging decisions in the future. The deutocerebrum processes information from the antennae, those sensitive appendages that detect odors and other chemical signals. For many insects, the sense of smell is paramount, guiding them to food sources, helping them find mates, and allowing them to recognize members of their own colony or species. The antennal lobes within the deutocerebrum contain structures called glomeruli, where signals from different odor receptors are sorted and processed before being sent to higher brain centers for interpretation. The tritocerebrum is the smallest of the three main brain regions and connects to the rest of the nervous system. It receives information from various sensory structures and helps coordinate motor responses. From the tritocerebrum, nerve cords extend down through the body, forming what is called the ventral nerve cord, a chain of ganglia that runs along the underside of the insect and serves as a secondary control center for the body's segments. Each segment of the insect body has its own ganglion, a cluster of nerve cell bodies that functions somewhat like a miniature brain. These ganglia can control local reflexes and movements without direct input from the head brain. This distributed nervous system allows for rapid responses to stimuli. When a cockroach detects air movement that might signal an approaching predator, the circle nerve cells at the rear of its body send signals directly to the leg ganglia, triggering an escape response in a matter of milliseconds, faster than signals could travel to the brain and back. Recently, scientists achieved a remarkable milestone in understanding insect brains. Researchers mapped the complete connectome of a fruit fly larva, creating a diagram of every neuron and every connection between neurons in the entire nervous system. This map revealed 3,016 neurons connected by 548,000 synapses. It was the first complete neural wiring diagram of an entire organism capable of complex behavior. The connectome showed how sensory information flows through the nervous system, how different brain regions communicate, and how circuits of neurons work together to produce behaviors like crawling, eating, and responding to threats. This wiring diagram revealed that the insect brain, despite its small size, exhibits organizational principles similar to those found in much larger brains. There are distinct pathways for processing different types of sensory information. There are feedback loops that allow the brain to adjust its own processing. There are hub neurons that connect many different regions, coordinating activity across the brain. The efficiency of this organization is extraordinary. Every neuron, every connection serves a purpose, contributing to the insect's ability to survive and thrive. The brain of an insect operates on remarkably low power. A bee's brain consumes about 10 microwatts of energy, roughly 1/10,000,000th the power used by a laptop computer. Yet it enables the bee to fly precisely, recognize flowers, navigate using the sun's position, remember locations, and communicate with nestmates through symbolic dances. This efficiency arises partly from the analog nature of neural computation in small brains, where signals can be graded rather than purely on-off, and partly from the compact physical arrangement that minimizes the distance signals must travel. Insects can learn and remember, demonstrating that their small brains are capable of plasticity, of changing in response to experience. Fruit flies can learn to avoid odors associated with unpleasant experiences and remember these associations for hours or even days. Bees can learn complex foraging routes, remembering the locations of profitable flowers and the times of day when those flowers produce the most nectar. Some species of wasps can recognize individual faces of other wasps, a cognitive feat that requires sophisticated visual processing and memory. The circadian clock, the internal timekeeper that regulates daily rhythms of activity and rest, is embedded within the insect brain. A small number of clock neurons, sometimes fewer than 100 cells, generate rhythmic patterns of gene expression and electrical activity that persist even in constant darkness. These clock neurons send signals throughout the brain and body, coordinating processes from metabolism to behavior with the 24-hour rotation of the Earth. In fruit flies, the clock neurons are located in several small clusters near the optic lobes. These cells express clock genes in rhythmic cycles, with some genes being active during the day and others at night. The proteins produced by these genes form feedback loops, where the presence of certain proteins inhibits the genes that produce them, creating an oscillation that takes approximately 24 hours to complete. This molecular clockwork is remarkably similar to the circadian mechanisms found in mammals, suggesting that the basic solution to timekeeping evolved early in animal history and has been conserved across widely separated lineages. The insect brain continuously monitors both internal and external conditions, adjusting the circadian clock to stay synchronized with the actual day-night cycle. Light detected by the eyes and by specialized photoreceptors in the brain itself provides the primary timing cue. Temperature also influences the clock, and insects have mechanisms to adjust their rhythms based on thermal conditions. This flexibility allows insects to adapt their activity patterns to seasonal changes and to different environments. Neurotransmitters, the chemical messengers that carry signals from one neuron to another, are present in insect brains in much the same way they are in vertebrate brains. Acetylcholine, dopamine, serotonin, and other familiar neurotransmitters all play important roles in insect neural function. Dopamine, for example, is involved in reward processing and learning. When a bee successfully finds nectar, dopamine signals in the brain reinforce the behaviors and associations that led to that success. Serotonin influences aggression and social behavior. Octopamine, a neurotransmitter more common in invertebrates, modulates arousal and energy mobilization. The fact that insects and vertebrates use similar neurotransmitters and similar neural architectures despite hundreds of millions of years of independent evolution suggests that there are fundamental constraints and optimal solutions in neural design. Certain molecules are particularly good at carrying signals across synapses. Certain circuit architectures are particularly effective at processing specific types of information. Evolution has discovered these solutions repeatedly in different lineages. Despite the power and sophistication of insect brains, they have limitations imposed by their small size. Complex computation requires neurons and connections, and there is only so much that can be packed into a tiny head. Insects excel at tasks that require rapid processing of sensory information and quick motor responses. They are adept at learning associations and patterns. But they generally do not exhibit the kind of abstract reasoning or long-term planning seen in large-brained vertebrates. Their intelligence is specialized, honed by evolution to solve the particular problems faced by small animals in complex environments. The study of insect brains continues to yield insights not only into how insects themselves function, but into general principles of neural computation. Because insect brains are small enough to be studied in their entirety, they serve as tractable models for understanding how networks of neurons give rise to behavior. The complete connectome of the fruit fly larva, for instance, allows researchers to trace exactly how sensory information flows through the nervous system to produce specific actions. These insights may eventually inform our understanding of larger brains, including our own, by revealing fundamental principles that scale across many orders of magnitude and size. When we observe an insect, we are watching decisions unfold, decisions made by a brain smaller than a grain of rice, yet capable of guiding its owner through a complex and changing world. The beetle navigating around obstacles, the butterfly selecting a flower, the ant following a pheromone trail. Each of these behaviors emerges from the coordinated activity of thousands or millions of neurons, from patterns of electrical and chemical signals flowing through circuits refined over millions of years of evolution. The insect brain is a reminder that intelligence comes in many forms, that sophisticated behavior does not require size, and that nature has found many ways to build a mind. How insects sleep and rest. The question of whether insects sleep might seem unusual at first. Sleep as we experience it involves a profound change in consciousness, a daily journey into oblivion from which we emerge restored. But sleep in its essence is a behavioral and physiological state characterized by reduced responsiveness to the environment, a preferred resting posture, and a rapid reversibility distinguishing it from other forms of inactivity. By these criteria, many insects do indeed sleep. Fruit flies, the workhorses of genetic research, have provided much of what we know about insect sleep. These tiny flies exhibit clear daily rhythms of activity and rest. During the night and during the heat of midday, fruit flies become quiescent. They find a suitable spot, often on the underside of a leaf or on the side of a container in the laboratory, and they remain there, still and unresponsive. Their reaction time to stimuli increases dramatically. A fly that would dart away from a puff of air when active will respond only sluggishly or not at all when in this resting state. This quiescence is not simply inactivity. Flies that are prevented from resting show signs of sleep deprivation. They require stronger stimuli to wake them from subsequent rest periods, suggesting a buildup of sleep pressure. They show compensatory rebound, sleeping more after a period of sleep deprivation, just as humans do after staying awake too long. Their cognitive performance declines when they are sleep deprived, demonstrating that this rest serves important restorative functions. The circadian clock plays a central role in regulating insect sleep. In fruit flies, clock genes control the timing of activity and rest periods, with certain genes promoting wakefulness during the day and others promoting sleep at night. The same molecular machinery that keeps circadian time also influences sleep drive. Mutations in clock genes can disrupt normal sleep patterns, causing flies to sleep at unusual times or to fragment their sleep into many short bouts rather than consolidating it into longer periods. Temperature interacts with the circadian system to influence insect activity and rest. Insects, being ectothermic, depend on environmental temperature for their body heat. Their metabolism, their neural function, and their behavior all vary with temperature. Fruit flies, for instance, show different sleep patterns at different temperatures. Cool temperatures promote rest, while warmer temperatures within the optimal range promote activity. However, extreme heat can induce a state of torpor, a protective inactivity that helps the insect avoid damage from thermal stress. The fruit fly brain contains sleep-promoting neurons, cells that become active during sleep and help maintain the sleeping state. These neurons integrate signals about circadian time, about the amount of prior waking activity, and about environmental conditions to determine whether the fly should be awake or asleep. When these neurons are artificially activated, they can induce sleep even in a fly that would normally be active. When they are silenced, sleep is reduced or fragmented. Honeybees also sleep, and their sleep is more complex than that of solitary insects. Young bees working inside the hive sleep in short bouts scattered throughout the day and night. Older forager bees, those that leave the hive to gather nectar and pollen, show more consolidated sleep, primarily at night. These foragers have been observed to sleep deeply enough that they can be gently touched or even picked up without waking. The antennae, those crucial sensory organs, droop during sleep, and brain activity measured through the hive wall shows patterns distinct from waking. Sleep in bees appears to be important for memory consolidation. Forager bees learn the locations of flowers, the landmarks surrounding the hive, and the quality of different food sources. Studies have shown that bees that are allowed to sleep after learning perform better on memory tests than bees that are sleep-deprived. This parallel with mammalian sleep and memory suggests that one of the fundamental functions of sleep, the processing and storage of information, is conserved across very different types of brains. Some insects show something resembling sleep even in their immature stages. Fruit fly larvae have periods of quiescence during which they do not feed or move, and from which they can be aroused by strong stimuli. Caterpillars, the larval stage of butterflies and moths, also show rest periods. These early life sleep states may serve different functions than adult sleep, perhaps related to growth and development rather than to learning and memory. Not all insects sleep in the same way or at the same times. Many moths and beetles are nocturnal, active at night and resting during the day. Their circadian clocks are phase-shifted relative to diurnal insects, with peak activity occurring after sunset. These nocturnal species must still solve the fundamental problem of when to be active and when to rest, and they do so using the same basic molecular and neural mechanisms, simply adjusted to a different phase relationship with the external light-dark cycle. Some insects inhabit environments where day and night are not clearly defined. Cave-dwelling insects living in perpetual darkness might be expected to lose their circadian rhythms. However, many retain functional clocks that continue to oscillate with a period close to 24 hours, even without light cues. These free-running rhythms suggest that the circadian clock serves functions beyond simply tracking day and night, perhaps coordinating internal physiological processes or allowing the insect to anticipate regular events in its environment, even when external cues are absent. Migratory insects face unique challenges in managing sleep and activity. Monarch butterflies, which migrate thousands of kilometers from North America to Mexico, must balance the need for sleep with the urgency of migration. During the migration period, monarchs fly during the day and rest at night, but their sleep may be reduced compared to non-migratory periods. The mechanisms that allow them to sustain activity during migration and whether they incur a sleep debt that must later be repaid remain topics of active research. Social insects like ants present fascinating questions about sleep in the context of colony life. In an ant colony, workers have different roles, some foraging outside, others tending the brood or maintaining the nest. These different roles may be associated with different sleep patterns. Some studies have found that ant workers sleep in brief bouts totaling several hours per day, but these bouts are not synchronized across the colony. At any given time, some workers are asleep while others are awake, ensuring that the colony as a whole remains functional around the clock. The queen ant and the workers may have different sleep needs. Queens, whose primary role is reproduction, may sleep more or differently than workers. In honeybee colonies, the queen's sleep has been observed to be deeper and longer than that of workers, perhaps because she does not face the same demands of foraging and nest maintenance. These variations in sleep across castes highlight how sleep is shaped by the ecological and social niche an individual occupies. Insect sleep is not always a passive state of simply turning off activity. Some insects engage in behaviors during sleep that suggest active processes are occurring. Bees sometimes move their antennae during sleep, and their brain activity shows patterns that could indicate dreaming or memory processing. Fruit flies twitch occasionally during sleep, much as mammals do during REM sleep. While we cannot know what, if anything, an insect experiences during sleep, these observations suggest that the sleeping brain is not simply idle. The evolution of sleep in insects raises intriguing questions. Sleep is nearly universal among animals, found in species from jellyfish to humans. The fact that insects, with their distinct evolutionary history and neural architecture, also sleep suggests that sleep serves some fundamental, unavoidable function. The leading hypotheses focus on brain maintenance, the clearance of metabolic wastes, the strengthening of important neural connections and the weakening of unimportant ones, and the restoration of energy reserves. All brains, it seems, require downtime to function optimally. The study of sleep in insects has practical applications as well. Understanding how the insect circadian system and sleep-wake cycles are regulated could lead to new methods for controlling pest species. If a pest insect's sleep could be disrupted, its fitness might decline, making it less able to damage crops or spread disease. Conversely, understanding how beneficial insects like pollinators regulate their sleep could help us protect them, perhaps by ensuring that pesticide applications avoid their sleep periods when they might be more vulnerable. As we consider the sleep of insects, we are reminded that even these small creatures require rest, that their tiny brains need time to consolidate memories, clear wastes, and prepare for the next day's challenges. The mosquito that lands on your arm at dusk has likely spent part of the day resting. The butterfly you see in the morning has emerged from a night of sleep. All around us, insects are engaging in this ancient behavior, this daily retreat into quiescence that restores and renews, making possible the activities of the waking world. The marvel of insect senses. To understand an insect's world, we must imagine perceiving reality through senses quite different from our own. While we rely heavily on binocular vision and hearing, insects often prioritize chemical detection and use visual systems constructed on entirely different principles. They taste with their feet, smell with their antennae, and see through eyes made of thousands of individual lenses. Their sensory world is rich and strange, tuned to the particular needs of small animals navigating complex environments. The compound eye is perhaps the most distinctive feature of insect vision. Rather than a single lens focusing light onto a retina as in vertebrate eyes, the compound eye consists of many individual units called ommatidia. Each ommatidium functions as a separate visual receptor with its own lens, crystalline cone, and photoreceptor cells. A fruit fly's eye contains about 760 ommatidia. A dragonfly's eye may contain 30,000. These ommatidia are arranged in a hemispherical array, giving many insects an extremely wide field of view, sometimes approaching 360 degrees. Each ommatidium points in a slightly different direction, so each samples light from a different part of the visual field. The insect's brain combines the signals from all ommatidia to create a mosaic image of the world. This image is not as sharp or detailed as what we see with our camera-type eyes, but it excels at detecting motion. When something moves across an insect's field of view, it sequentially triggers ommatidia, creating a clear signal of movement that can be processed extremely quickly. This is why it is so difficult to swat a fly. The fly's visual system detects the approaching hand almost instantly, and its nervous system triggers an escape response faster than our hand can complete its motion. Dragonflies, which are aerial predators, have some of the most sophisticated compound eyes in the insect world. Their eyes occupy most of their head, providing nearly all-around vision. Different regions of the eye have different resolutions, with higher density of ommatidia in the forward-looking dorsal region used to spot prey against the sky. Dragonflies can see a wider spectrum of colors than humans, including ultraviolet light, and they can distinguish polarization patterns in light, which helps them navigate and may help them detect water surfaces. Many insects can indeed perceive ultraviolet light, which is invisible to us. Flowers often have patterns visible only in UV, patterns that guide insects to nectar and pollen. What appears to our eyes as a plain yellow flower might appear to a bee as a flower with bold UV markings pointing toward its center. This ultraviolet vision is not a curious extra sense, but a fundamental part of how many insects interact with flowers. Plants and their insect pollinators have co-evolved, with plants developing UV patterns that attract pollinators and pollinators developing the ability to see those patterns. Some insects have simple eyes in addition to compound eyes. These simple eyes, called ocelli, are typically 3 in number, arranged in a triangle on top of the head. Ocelli cannot form images, but they are sensitive to light intensity and to the horizon, helping the insect maintain stability during flight. They provide rapid feedback about orientation, supplementing the more detailed but slower information from the compound eyes. The antennae are the primary olfactory organs of insects. These remarkable appendages are covered with sensory structures called sensilla, each containing receptor neurons sensitive to specific chemical compounds. A moth's feathery antennae can detect pheromone molecules at concentrations of a few hundred molecules per cubic centimeter of air. A male moth can detect and track the pheromone plume of a female from distances of several kilometers, flying upwind toward the source of the scent in complete darkness. The diversity of antennal shapes reflects the different olfactory needs of different species. Beetles that feed on rotting wood have stout, clubbed antennae. Butterflies have long, thin antennae with knobbed tips. Male mosquitoes have bushy antennae covered with fine hairs that detect the characteristic wing beat frequency of females, allowing them to locate mates by sound. Each shape represents an optimization for a particular sensory task. Insects also taste through specialized sensors called contact chemoreceptors. These are located not only on mouth parts where we might expect them, but also on the feet. A butterfly landing on a leaf can taste the leaf's surface through receptors on its feet, determining in an instant whether this is a suitable plant on which to lay eggs. Flies taste food sources through their feet as well, which is why a fly walking across your food is not merely investigating visually but actively sampling with every step. Touch and mechanoreception are crucial senses for insects. Insects have sensory hairs called setae distributed across their body surface. These hairs can detect air currents, vibrations, and physical contact. The cerci at the rear of a cockroach's body are covered with sensory hairs that detect even the slightest air movement, providing an early warning system for approaching predators. When you try to step on a cockroach, the displaced air from your descending foot can trigger its escape response before contact is made. Many insects are sensitive to vibrations transmitted through solid substrates. Tree hoppers communicate by sending vibrations through plant stems, These signals can travel many centimeters through the plant and are detected by specialized organs in the legs. Different vibrational signals convey different meanings, allowing treehoppers to locate mates, warn of predators, or claim territory, all without making airborne sounds that might attract the attention of predators. Some insects can detect magnetic fields, though the mechanisms remain incompletely understood. Monarch butterflies use the Earth's magnetic field as one of several cues for navigation during migration. Magnetoreceptors might be located in the antenna or might involve chemical reactions in the eyes that are influenced by magnetic fields. This sense, combined with the ability to use the sun's position as a compass and to recognize visual landmarks, allows monarchs to navigate with remarkable precision across vast distances. Temperature and humidity sensors help insects find suitable microhabitats and avoid dangerous extremes. Mosquitoes use temperature sensors to detect the warmth of a potential host's skin. Certain beetles that feed on recently burned wood can detect infrared radiation from fires, allowing them to find fresh burn sites from tens of kilometers away. These beetles arrive at forest fires within hours, ready to lay eggs in the dead and dying trees before other insects can claim this resource. Hearing in insects is accomplished through a variety of structures, not a single type of ear. Crickets and katydids have tympanic organs on their front legs, thin membranes that vibrate in response to sound. Moths have tympanic organs on their thorax or abdomen tuned specifically to detect the ultrasonic echolocation calls of bats, their primary predators. When a moth's ear detects a bat call, the moth immediately initiates evasive flight maneuvers, diving or turning erratically to avoid capture. Mosquitoes detect sound through their antennae. The antennal shaft vibrates in response to sound waves, and this vibration is detected by specialized sensory cells at the base. Male mosquitoes are particularly sensitive to frequencies around 400 to 600 Hz, which corresponds to the wing beat frequency of females. In the swarms that form at dusk, males use this auditory sense to locate females for mating. Some insects can detect electric fields. Bees, with their fuzzy bodies, accumulate positive charge as they fly through the air. Flowers often have a slight negative charge relative to the surrounding air. When a bee approaches a flower, the electric field around the flower changes, and the bee can detect this change through sensory organs at the base of mechanosensory hairs. This electric sense might help bees identify which flowers have recently been visited by other bees and are therefore likely to have less nectar. The integration of all these senses occurs in the insect brain, where streams of information from eyes, antennae, taste receptors, touch sensors, and other modalities are combined to create a unified perception of the environment. This multimodal integration allows insects to make rapid decisions. A foraging bee uses vision to approach a flower from a distance, switches to olfaction to identify the flower species when closer, uses touch to land, and uses taste to evaluate the nectar. All of this happens in seconds, guided by a brain smaller than a pinhead. The sensory world of an insect is not merely a dimmer or simpler version of our own. It is qualitatively different, built around different priorities and constraints. An insect lives at a different scale, where surface tension of water is a powerful force, where a breeze is a gale, where a raindrop is a massive projectile. The senses of insects are exquisitely tuned to this scale, providing the information needed to survive and thrive in a world we can observe but never truly experience as they do. When we watch a bee visiting flowers, we see only the visible light. We smell only the strong scents. We feel only the obvious textures. The bee experiences UV patterns, detects subtle volatile compounds we cannot perceive, feels electric fields we never sense. Its world is richer in certain dimensions than ours, even as it lacks dimensions we take for granted. This humbling realization that other minds perceive realities inaccessible to us expands our understanding of what it means to sense and to know. Breathing without lungs. Insects accomplish one of life's most essential tasks, the exchange of oxygen and carbon dioxide, without lungs, without blood that carries oxygen, and without a circulatory system dedicated to gas transport. Instead, they employ a network of tubes that penetrates throughout their body, delivering oxygen directly to tissues and removing carbon dioxide in a system as different from our own as it is effective for creatures of small size. This system is called the tracheal system, and its fundamental components are the tracheae, tubes that begin at openings on the body surface called spiracles and branch repeatedly as they extend inward. The walls of tracheae are lined with chitin, the same material that forms the exoskeleton, arranged in spiral thickenings that give the tubes structural support while maintaining flexibility. As the tracheae branch, they become narrower, eventually forming trocheoles, the finest tubes with diameters measured in microns that terminate directly on or within cells. Spiracles are the gateways of this system. These openings are distributed along the sides of the insect body, typically 1 or 2 pairs per segment. A moth might have 8 or 9 pairs of spiracles. A beetle has a similar number. The spiracles are not simply holes. They are controlled openings equipped with valves that can close to regulate gas exchange and, crucially, to prevent water loss. Water conservation is a significant challenge for small terrestrial animals. The surface area to volume ratio is high, meaning that relative to their mass, insects have a lot of surface area from which water can evaporate. Open spiracles would allow water vapor to escape along with carbon dioxide. Insects solve this problem through active control of spiracle opening. The spiracles open only when necessary for gas exchange and remain closed or partially closed much of the time, reducing respiratory water loss. This control of spiracle opening is not random but follows specific patterns. Many insects exhibit discontinuous gas exchange, a cycle in which spiracles are tightly closed for a period, then flutter open and closed rapidly, and finally open fully for a brief time before closing again. During the closed phase, oxygen is consumed from the tracheal system and carbon dioxide builds up, but the carbon dioxide dissolves in the body fluids rather than accumulating as gas. During the flutter phase, spiracles open just enough to allow a small amount of oxygen in. During the open phase, spiracles open wide, fresh air rushes in, and the accumulated carbon dioxide is expelled. This cycle can repeat every few minutes or hours depending on the insect's metabolic rate and environmental conditions. The tracheal system works primarily through diffusion. Oxygen moves down its concentration gradient from the outside air where it is abundant, through the spiracles, along the tracheae, and into the tracheoles where it is consumed by respiring cells. Carbon dioxide moves in the opposite direction, from high concentration in the tissues to low concentration in the outside air. For very small insects, diffusion alone is sufficient to meet all respiratory needs. Larger or more active insects supplement diffusion with active ventilation. They contract and expand their abdomen rhythmically, compressing and expanding the tracheal system to pump air in and out. Flying insects, which have extraordinarily high metabolic rates, use this ventilation extensively. The flight muscles of a bee or a dragonfly consume oxygen at a rate comparable to that of a flying bird or bat when corrected for body mass, and this demand is met by vigorous pumping of air through the tracheal system. Some insects have air sacs, expandable regions of the tracheal system that serve as reservoirs and help with ventilation. These sacs can be compressed during flight or other activity, driving air through the system more forcefully. The air sacs also reduce the insect's overall density, which may be beneficial for flight. The tracheal system imposes constraints on insect size. Because oxygen must diffuse through the tracheae to reach deep tissues, there is a limit to how large an insect can be while still getting adequate oxygen to all its cells. This is one reason why the giant insects of the Carboniferous period required higher atmospheric oxygen levels. With more oxygen in the air, diffusion gradients were steeper, allowing oxygen to penetrate further into large bodies. In aquatic insects, the tracheal system faces different challenges. Some aquatic insects come to the surface to breathe, extending a breathing tube or siphon through the water surface to access air. Mosquito larvae are a familiar example, hanging from the surface film with their siphon breaking the surface. Other aquatic insects carry air with them underwater, trapping it as a bubble or within a layer of water-repellent hairs. This air bubble serves as a temporary oxygen supply, but it also functions as a physical gill. As the insect consumes oxygen from the bubble, oxygen from the surrounding water diffuses into the bubble to equalize the concentration. This allows the bubble to last longer than if it were simply a fixed store of gas. Some aquatic insects have tracheal gills, thin-walled extensions of the body that contain tracheal branches. Oxygen dissolved in the water diffuses across the gill wall into the tracheae. Mayfly nymphs have delicate tracheal gills along their abdomen. Damselfly nymphs have 3 leaf-like gills at the end of their abdomen. These structures increase the surface area for gas exchange and allow the insect to extract oxygen directly from the water without coming to the surface. The efficiency of the insect respiratory system is remarkable. It delivers oxygen without the need for respiratory pigments like hemoglobin. Most insects do not use their circulatory fluid to transport oxygen at all. The hemolymph, the fluid that bathes insect tissues, primarily transports nutrients, hormones, and immune cells, not oxygen. This separation of circulatory and respiratory functions is a fundamental difference between insect and vertebrate physiology. The tracheal system must be renewed periodically. When an insect molts, shedding its old exoskeleton to grow, it also sheds the chitinous lining of its tracheae. Before each molt, new cuticle forms inside the old, including new tracheal lining. After the molt, the old tracheal lining is pulled out through the spiracles, a delicate process that must be completed successfully for the insect to breathe normally. Any failure in this process can be fatal. In some insects, parts of the tracheal system are fluid-filled at certain stages of development. This may serve various functions, including providing hydraulic pressure to extend structures like wings after a molt, or managing the transition from aquatic larval stages to terrestrial adult stages. The ability to control whether portions of the tracheal system are air-filled or fluid-filled demonstrates the flexibility of this respiratory architecture. The tracheal system interconnects with many other aspects of insect physiology. The need to protect tracheae from water while allowing gas exchange influences insect behavior and habitat selection. The limitations of tracheal oxygen delivery influence body size and shape. The metabolic rate supportable by the tracheal system constrains activity levels and ecological roles. And the tracheal system's efficiency at small scales is one reason why insects have been so successful as a group, able to occupy niches that larger animals cannot exploit. As we breathe in and out, our lungs expand and contract. Our heart pumps oxygenated blood throughout our body, a complex system of interdependent parts. An insect achieves the same goal through a system of branching tubes, through spiracles that open and close, through diffusion and sometimes ventilation, all without our central pumping heart, without lungs, without hemoglobin. It is a solution to the problem of respiration that works beautifully at the scale insects inhabit, a reminder that life has discovered many ways to accomplish essential tasks. The exoskeleton and insect strength. The body of an insect is enclosed and supported not by an internal skeleton of bone, but by an external skeleton, an exoskeleton that forms the outer boundary of the organism. This exoskeleton serves multiple functions simultaneously. It is protective armor, structural support, a barrier against water loss, and the framework to which muscles attach. It is one of the key innovations that has made insects so successful. The exoskeleton is composed primarily of chitin, a long-chain polymer of a sugar derivative. Chitin molecules are arranged in crystalline arrays and embedded in a matrix of proteins, creating a composite material similar in concept to fiberglass or carbon fiber. This chitin protein composite, called cuticle, has properties that can be tuned over a wide range. By varying the type and amount of proteins, by altering the arrangement of chitin fibers, and by adding other compounds, insects create cuticle with properties ranging from hard and rigid to soft and flexible. The hardest parts of the exoskeleton, such as a beetle's wing covers, are heavily sclerotized. Sclerotization is a tanning process in which proteins in the cuticle are cross-linked through chemical reactions, creating a tough, rigid material that resists deformation and protects against predators, parasites, and physical damage. Sclerotized cuticle can be remarkably hard. Some beetles have such tough exoskeletons that they are difficult to pierce even with a steel pin. Other parts of the exoskeleton remain soft and flexible. The membranes between body segments, the joints of the legs, the thin cuticle that allows the abdomen to expand when an insect feeds, all these areas are made of cuticle with little or no sclerotization. This flexibility is essential for movement. Muscles cannot work without joints that can bend, and the exoskeleton provides those joints through regions of controlled flexibility. Beneath the tough outer layer of cuticle is a thicker inner layer with different properties. The exoskeleton is thus a multi-layered structure, with each layer contributing different properties to the whole. The outermost layer is often covered with a waxy coating, the epicuticle, which is crucial for waterproofing. This waxy layer is extremely thin, just a few molecules thick in some places, but it makes the difference between life and death for a terrestrial insect. Without it, the insect would desiccate rapidly. The exoskeleton must accommodate growth, and insects grow by molting, a process called ecdysis. The old exoskeleton cannot expand, so periodically the insect must shed it entirely and grow a new, larger one. Before molting, the insect secretes a new cuticle beneath the old one. Then it releases enzymes that digest the inner layers of the old cuticle, recycling the materials. The insect absorbs water or swallows air to expand its body, splitting the old exoskeleton along predetermined lines of weakness. It then crawls out of the old exoskeleton, expands the new soft cuticle to its full size, and waits for it to harden. During this vulnerable period, the insect is soft, pale, and defenseless. Many predators specialize in finding and eating insects immediately after molting. The number of molts varies among species. Some insects molt 4 or 5 times during their larval stage. Others molt dozens of times. Each molt represents a period of risk, but also an opportunity to repair damage, replace lost appendages, and grow to a new size. The timing of molts is controlled by hormones, primarily ecdysone, which coordinates the complex sequence of events required for successful molting. The exoskeleton allows insects to achieve extraordinary strength relative to their size. The mechanical advantage provided by an external skeleton with muscles attached on the inside creates leverage ratios that favor force over distance. A leafcutter ant can carry pieces of leaf weighing up to 50 times its own body weight. A rhinoceros beetle can support more than 800 times its body weight on its back. These feats are possible partly because of scaling laws. Strength scales with the cross-sectional area of muscles, which scales with the square of linear dimensions. Body weight scales with volume, which scales with the cube of linear dimensions. As animals get smaller, strength-to-weight ratio increases. An ant is not strong because it has super muscles, but because the physics of scaling work in its favor. The exoskeleton also creates challenges. All muscles are on the inside, attached to internal surfaces of the cuticle. This places constraints on how much muscle mass an insect can have, because there is only so much internal surface area for attachment. As insects get larger, the relative amount of internal surface area decreases, eventually limiting how large an insect can be and still have enough muscle to move its body. The attachment of muscles to the inside of the exoskeleton creates the joints and leverage systems that allow insects to move. At each joint, opposing muscles attach on either side of a flexible membrane. When one muscle contracts, it bends the joint. When the opposite muscle contracts, the joint bends the other way. This arrangement is found in every insect limb, in every movable body part. The precision with which these muscles are controlled allows insects to perform delicate manipulations with their legs and mouth parts. Wings are extensions of the exoskeleton, stiffened by hollow veins that contain tracheal branches and nerves. The wing membrane itself is 2 layers of cuticle pressed together, incredibly thin and light, yet strong enough to generate the forces needed for flight. The veins provide structural support, creating a framework that resists bending and twisting during the wing beat cycle. Different patterns of veins create different flight characteristics. Dragonflies have many cross veins, creating a fine meshwork that provides great stiffness. Butterflies have fewer veins and their wings are more flexible, suited for a different style of flight. The colors we see on insects are often products of the exoskeleton. Some colors come from pigments embedded in the cuticle, molecules that absorb certain wavelengths of light and reflect others. Other colors, especially iridescent blues and greens, are structural colors produced by microscopic structures in the cuticle that interfere with light waves. The brilliant blue of a morpho butterfly's wings comes from scales with layered structures that reflect blue wavelengths, while allowing other wavelengths to interfere destructively and disappear. Structural colors can be extraordinarily pure and bright, and they do not fade over time as pigments might. Some insects have exoskeletons with specialized structures for sound production. Crickets rub a scraper on one wing against a file on the other wing, a process called stridulation, producing their characteristic chirping. The frequency of the chirp is determined by the spacing of teeth on the file, and the volume is amplified by regions of wing membrane that vibrate like a drumhead. Each species of cricket has its own song, a consequence of slight differences in file structure and wing mechanics. The exoskeleton also provides attachment points for sensory structures. Every hair, every sensory pit, every compound eye is part of or attached to the exoskeleton. The cuticle itself can be a sensory structure, with nerve endings that detect strain and deformation, providing proprioceptive feedback about the position and movement of body parts. Despite its many advantages, the exoskeleton imposes limits. It must be shed to allow growth, making the insect vulnerable. It adds weight that must be carried. It reduces flexibility in some dimensions while providing it in others. Yet the exoskeleton has proven to be a highly successful solution to the problems of structure and protection, one that has served insects well for 400 million years. In every beetle that crawls, every bee that flies, every grasshopper that leaps, we see the exoskeleton in action, bending at joints, supporting wings, protecting internal organs, a framework that makes possible the complex behaviors and diverse forms of the insect world. Life cycles and metamorphosis. The life of an insect often unfolds in distinct stages, each with its own form, its own ecology, its own role in the species' survival strategy. This transformation from one stage to another, called metamorphosis, is one of the most remarkable features of insect biology. It allows a single species to exploit different resources at different life stages, reducing competition between juveniles and adults and often producing forms so different that early naturalists classified them as separate species. Insects are divided into two broad groups based on their development: those that undergo incomplete metamorphosis and those that undergo complete metamorphosis. Incomplete metamorphosis, or hemimetabolous development, is the ancestral condition. In these insects, the young, called nymphs, resemble small versions of the adults. They hatch from eggs, grow through a series of molts, gradually increasing in size and developing wing buds that become functional wings in the final molt to adulthood. Grasshoppers, dragonflies, and true bugs are examples of insects with incomplete metamorphosis. A grasshopper nymph emerging from its egg looks like a tiny wingless grasshopper. It has the same basic body plan as the adult, the same feeding habits, and often lives in the same habitat. With each molt, it grows larger, its wing buds become more prominent, and its proportions shift slightly. After perhaps 5 or 6 molts, it reaches adult size, its wings expand and harden, and it becomes capable of flight and reproduction. The transition from nymph to adult is gradual, a matter of degree rather than kind. Complete metamorphosis, or holometabolous development, represents an evolutionary innovation that has contributed greatly to insect diversity. More than 80% of all insect species undergo complete metamorphosis, including beetles, butterflies, moths, flies, bees, wasps, and ants. In these insects, the life cycle has 4 distinct stages: egg, larva, pupa, and adult. Each stage is so different from the others that they might seem to be unrelated organisms. The larva is a feeding machine, devoted almost entirely to eating and growing. Caterpillars, grubs, and maggots are all larval forms of different insect groups. The larva typically has a simple body structure, often with chewing mouth parts well suited for consuming large quantities of food. It lacks wings and often has limited sensory capabilities compared to the adult. The larva's job is to accumulate resources, to store energy and nutrients that will later fuel metamorphosis and adult activity. Larvae grow through a series of molts, each larval stage called an instar. A caterpillar might pass through 5 or 6 instars, growing larger with each one. Some caterpillars increase their body weight by several thousand times from hatching to final instar. This enormous growth is possible because the larva is a specialized feeding stage, unencumbered by the complexities of flight or reproduction. When the larva has accumulated sufficient resources and reached the appropriate size, it enters the pupal stage. The pupa is one of nature's most extraordinary life stages. Externally, the pupa appears inactive, a dormant capsule that shows little sign of life. But internally, a radical transformation is underway. Most of the larval tissues break down through programmed cell death and are reorganized into adult structures. Wings form, compound eyes develop, the nervous system is rewired, reproductive organs mature. The adult insect is built from the raw materials of the larva. This process is guided by cascades of gene activation. Groups of cells called imaginal discs, which have been present in the larva since embryonic development but remained undeveloped, now proliferate and differentiate, forming the adult structures. The transformation is total. A caterpillar that crawled and chewed leaves becomes a butterfly that flies and sips nectar. A grub that lived in soil becomes a beetle that might fly or swim. The pupa is a life stage dedicated to reconstruction. The duration of the pupal stage varies widely. Some butterflies pupate for just a week or 2. Others, especially those that overwinter as pupae, remain in this stage for months. The timing is controlled by environmental cues like temperature and day length, as well as by internal developmental programs. When metamorphosis is complete, the adult insect emerges, a process called eclosion. Eclosion is a delicate and critical moment. The adult must break out of the pupal case, expand its wings, and allow its exoskeleton to harden before it can function. A butterfly emerging from its chrysalis is soft and vulnerable. It pumps hemolymph into its wing veins, expanding the wings to their full size. It must hang quietly while the wings dry and harden, a process that can take hours. During this time, it cannot fly or escape from predators. Many adult insects never make it past eclosion, falling victim to predators or adverse conditions at this vulnerable moment. Once the adult form is achieved, there is no more molting, no more growth. Adult insects have reached their final size. Their primary goals now are to find mates, reproduce, and in some species to disperse to new habitats. The adult stage may last days, weeks, or even years, depending on the species. The adaptive advantages of complete metamorphosis are significant. Larvae and adults can exploit entirely different resources. Caterpillars eat leaves while butterflies drink nectar. Mosquito larvae filter feed in water while adults fly and feed on blood or nectar. This division of labor reduces competition between life stages. It also allows each stage to specialize. The larva can evolve structures optimized for feeding and growth without the constraints of needing to fly or reproduce. The adult can evolve structures optimized for dispersal and reproduction without the constraints of needing to process large amounts of food. Different insect groups have different larval forms reflecting their different ecologies. Beetle larvae, called grubs, often have well-developed legs and chewing mouthparts. They might be predators, scavengers, or plant feeders. Fly larvae, called maggots, often lack distinct legs and have reduced heads. Many are adapted for life in fluids or decaying organic matter. Butterfly and moth larvae, caterpillars, are specialized for eating plant material and have specialized mouthparts and often striking colors or patterns for defense. Some insects have aquatic larvae and terrestrial adults, allowing them to exploit both environments. Dragonflies, mayflies, and caddisflies spend most of their lives as aquatic nymphs or larvae, then emerge as flying adults that may live for just a few days or weeks. The mosquito's life cycle similarly bridges water and air, with larvae and pupae in water and adults in air. This life cycle diversity allows insects to utilize resources unavailable to organisms confined to a single environment. The timing of metamorphosis and adult emergence is often synchronized within populations, leading to mass emergences. Periodical cicadas spend 13 or 17 years underground as nymphs, feeding on root sap, then emerge all at once in enormous numbers. This synchrony satiates predators. There are so many cicadas emerging that predators cannot possibly eat them all, ensuring that many survive to reproduce. The triggers for such mass emergences involve complex environmental cueing, with nymphs somehow counting years or seasonal cycles underground. Hormones regulate metamorphosis and molting. Juvenile hormone keeps insects in larval or nymphal stages. When juvenile hormone levels decline, metamorphosis proceeds. Ecdysone triggers molting. The interplay between these hormones and others determines the timing and nature of developmental transitions. Environmental factors like temperature, nutrition, and day length influence hormone levels, linking development to external conditions. In some insects, metamorphosis can produce different adult forms depending on environmental conditions. Aphids can produce winged or wingless adults depending on population density and food availability. Some ants produce different castes, workers, soldiers, and queens from genetically identical larvae by varying nutrition during development. These cases demonstrate that metamorphosis is not a rigid program but a flexible process that can produce different outcomes in response to circumstances. The study of metamorphosis has provided profound insights into developmental biology. How does a caterpillar's cell know to become part of a butterfly's wing? How are genes activated in the right sequence to build complex structures? These questions have been addressed through research on insects, particularly fruit flies, whose metamorphosis can be manipulated and observed in the laboratory. The answers have implications far beyond insects, revealing fundamental principles of how genes control development in all organisms. As we observe a butterfly visiting flowers or a beetle crawling across bark, we are seeing only one stage of a complex life cycle. Behind the adult lies a history of transformation, of a larva that ate and grew, of a pupa that rebuilt its body from the inside out. The insect before us is a product of metamorphosis, a testament to the developmental flexibility and evolutionary innovation that have made insects the most diverse group of animals on Earth. Communication in the insect world. Insects live in a world of signals, of messages sent and received through channels we rarely perceive. They communicate through chemicals that drift on the air, through sounds pitched too high or too low for our ears, through touches and vibrations, through visual displays. This communication allows insects to find mates across great distances, to coordinate activities within colonies of thousands or millions, to warn of danger and to claim territory. The languages of insects are ancient and sophisticated, refined over millions of years of evolution. Chemical communication dominates the insect world. Pheromones are chemical signals produced by one individual that affect the behavior or physiology of other individuals of the same species. A single molecule of the right pheromone detected by the right receptor can trigger complex behavioral responses. The diversity of pheromones is staggering. There are sex pheromones that attract mates, alarm pheromones that warn of danger, trail pheromones that mark paths to food, aggregation pheromones that bring individuals together, and many others. Sex pheromones are among the most studied. Female moths release pheromones from specialized glands, typically located at the tip of the abdomen. These pheromones are species-specific blends of volatile compounds, often long-chain alcohols, aldehydes, or acetates. The pheromone molecules evaporate and are carried downwind, forming a plume that spreads and dilutes as it moves away from the source. A male moth flying through this plume detects the molecules with receptors on his antennae. The sensitivity of this detection is extraordinary. A male silkworm moth can respond to just a few hundred molecules of the sex pheromone bombicol. His feathery antennae are covered with thousands of sensory hairs, each containing receptor cells tuned specifically to bombicol. When bombicol molecules bind to these receptors, they trigger electrical signals that travel to the brain. The male begins flying upwind, zigzagging back and forth to stay within the plume. If he loses the scent, he casts about, flying in expanding loops until he relocates the plume. By repeating this process, orienting upwind whenever he detects pheromone, he can track the plume back to its source, sometimes from distances of several kilometers. The specificity of pheromones allows many species to coexist without confusion. Different species use different pheromone compounds or different ratios of the same compounds. A male moth is exquisitely sensitive to his own species' pheromone, but unresponsive to the pheromones of even closely related species. This chemical isolation is one of the mechanisms that keep species separate, preventing hybridization and maintaining distinct evolutionary lineages. Social insects use pheromones extensively to organize colony activities. An ant colony functions as a superorganism, with thousands of individuals cooperating to achieve goals no single ant could accomplish. Much of this coordination is achieved through pheromones. When a foraging ant finds food, she returns to the nest laying a trail pheromone from a gland in her abdomen. Other ants encountering this trail are stimulated to follow it, and if they also find food, they reinforce the trail with their own pheromone. The trail grows stronger as more ants use it, creating a positive feedback loop that recruits many workers to a rich food source. Trail pheromones are typically volatile compounds that evaporate within minutes or hours. This volatility is adaptive. If a food source is depleted, ants stop reinforcing the trail, and the pheromone evaporates, causing the trail to disappear. The colony thus automatically redirects its foraging effort from exhausted sources to fresh ones. This simple system, with no central planning or decision-making, achieves efficient resource exploitation through the collective behavior of many individuals following simple rules. Alarm pheromones coordinate defensive responses. When a honeybee stings a threat, she releases alarm pheromone from glands near the sting. This pheromone smells sweet to humans, reminiscent of banana or pear, but to other bees it is a signal of danger. Bees near the source become agitated and aggressive, more likely to sting. If many bees sting, the concentration of alarm pheromone increases, recruiting even more bees to the defense. This amplification can turn a few defensive stings into a mass attack, a response that deters even large predators. Queen pheromones regulate reproduction in social insect colonies. The queen honeybee produces a pheromone called queen mandibular pheromone that inhibits the development of ovaries in worker bees and suppresses their instinct to raise new queens. As long as this pheromone is present throughout the hive, workers remain sterile and loyal to the existing queen. If the queen dies or becomes weak, the pheromone concentration declines and workers begin raising new queens from young larvae. This chemical control of reproduction maintains colony cohesion and prevents internal conflict. Acoustic communication, the use of sound, is employed by many insects. Crickets and katydids are perhaps the most familiar sound-producing insects. Males sing by rubbing their wings together, a behavior called stridulation. One wing has a file, a row of tiny teeth, and the other has a scraper, a hardened edge. When the wings are rubbed together, the scraper catches on each tooth in sequence, producing a series of rapid impacts that create vibrations. These vibrations are amplified by specialized areas of the wing membrane that act as resonators. Each species of cricket has its own song, characterized by particular frequencies, rhythms, and patterns. Some produce continuous trills, others make chirps with specific numbers of pulses. The song serves to attract females and to space out males, with each male establishing a territory centered on his singing perch. Females listen to the songs and move toward males whose songs they find attractive. Exactly what makes a song attractive varies among species, but it often involves features like loudness, frequency, and consistency of rhythm. The temperature affects cricket song. Crickets are cold-blooded, and their metabolic rate, including the rate at which muscles can contract, depends on temperature. As temperature increases, crickets sing faster. This relationship is so reliable that you can estimate the temperature by counting cricket chirps. For some species, the formula is to count the number of chirps in 14 seconds and add 40, giving the temperature in degrees Fahrenheit. This quirk of physiology has made crickets into living thermometers. Cicadas produce some of the loudest sounds in the insect world. Males have specialized sound-producing organs called tymbals on the sides of their abdomen. Each tymbal is a ribbed membrane that can be buckled in and out by muscles, rather like clicking the metal lid of a jar in and out. When the tymbal buckles, it produces a click. By rapidly buckling the tymbals in and out and by using air sacs in the abdomen as resonators, cicadas create their characteristic buzzing or whining songs. Some species are loud enough to cause hearing damage in humans at close range. Mosquitoes communicate acoustically during mating swarms. Males and females both produce sounds with their wings as they fly, but at different frequencies. Males adjust their wing beat frequency to harmonize with the frequency of nearby females, creating a shared tone. This acoustic coupling helps males locate females in the chaotic environment of a swarm and may serve as a courtship signal. The mosquito's antennae, sensitive to sound vibrations, detect these wing tones and guide the insects toward each other. Vibrational communication, distinct from airborne sound, involves signals transmitted through solid substrates. Many insects communicate by vibrating plant stems, leaves, or the ground. These vibrations can travel considerable distances through plants, sometimes farther than airborne sounds could travel. Tree hoppers, small plant-feeding insects, send complex vibrational signals through the plants they inhabit. Males produce courtship signals, females respond, and the resulting duets help the sexes find each other for mating. Stink bugs also use vibrational communication. Males produce substrate-borne vibrations by shaking their abdomen, and females respond with their own signals. The insects can localize the source of vibrations by comparing the intensity and timing of vibrations arriving at different legs, allowing them to walk directly toward a signaling partner even though they cannot see each other. Visual communication is important for insects with good vision. Fireflies, beetles that can produce light through bioluminescence, use flashing patterns to find mates. Each firefly species has its own flash pattern, characterized by the duration of flashes, the interval between flashes, and the color of the light. Males fly over fields at dusk, flashing their species-specific pattern. Females perched in the vegetation watch for the correct pattern and respond with their own flash. The male flies toward the responding female, and the two engage in a dialogue of flashes that culminates in mating. The light is produced by a chemical reaction in specialized cells in the firefly's abdomen. A molecule called luciferin is oxidized by the enzyme luciferase in the presence of oxygen and ATP, producing light with very little heat. The firefly controls the flashing by regulating oxygen delivery to the light-producing cells. The efficiency of this bioluminescence is remarkable, with nearly 100% of the energy going into light rather than heat, far more efficient than any artificial light source. Some female fireflies of certain species have evolved to exploit the communication system of other species. These femme fatale fireflies can mimic the flash responses of females of other species. When a male of the other species approaches expecting to mate, the predatory female captures and eats him. This aggressive mimicry demonstrates how communication systems can be exploited and how evolution produces countermeasures and countermeasures in an ongoing arms race. Butterflies use visual signals for mate recognition and courtship. Many butterflies are brightly colored, and these colors serve as species recognition cues. Males often have ultraviolet patterns on their wings that are visible to other butterflies, but not to us. During courtship, butterflies may fly in elaborate patterns, displaying their wing colors and patterns to potential mates. The combination of color, pattern, and flight behavior ensures that mating occurs between individuals of the same species. Bees communicate the location of food sources through a remarkable behavior called the waggle dance. A forager bee that has found a rich source of nectar returns to the hive and performs a dance on the vertical surface of the comb. The dance is a figure-8 pattern with a waggle run in the middle where the bee moves forward while waggling her abdomen from side to side. The direction of the waggle run, relative to vertical, indicates the direction of the food source relative to the sun. The duration of the waggle run indicates the distance to the food source. Bees following the dancer can extract this information and fly directly to the food source, even though they have never been there before. This dance language is symbolic. The angle on the comb represents an angle in the landscape. The duration of a behavior represents a distance. These are abstract representations, a form of communication that approaches language in its complexity. The discovery of the waggle dance by Carl von Frisch was one of the most significant findings in animal behavior. demonstrating that insects are capable of communicating detailed information about the world. Tactile communication through direct contact is also important. Ants touching antennae exchange information about colony identity and perhaps about individual identity. This antennal contact involves the exchange of cuticular hydrocarbons, waxy compounds on the surface of the exoskeleton that serve as chemical signatures. By reading these signatures, ants can distinguish nestmates from non-nestmates, allowing them to cooperate with relatives while excluding or attacking strangers. Bees and wasps also use antennal contact as part of their social interactions. When a forager bee returns to the hive, she may be contacted by other bees who assess the pheromones and odors on her body, gaining information about the flowers she has visited. This olfactory information can influence the foraging choices of the bees receiving it, spreading knowledge of good food sources through the colony. The communication systems of insects are not static but evolve in response to environmental pressures and to the communication systems of other species. In noisy acoustic environments, insects may shift the frequency of their calls to avoid interference. Pheromone signals may evolve to become more or less volatile depending on the typical distances over which communication occurs. Visual signals may be tuned to match the sensory capabilities of intended receivers while remaining inconspicuous to predators. As we listen to the evening chirping of crickets, watch fireflies blinking in the summer dusk, or observe ants following invisible trails, we are witnessing communication in action. These insects are sending and receiving messages, coordinating their behavior, finding mates, warning of danger, sharing information. The signals they use are often imperceptible to us, existing in frequencies we cannot hear, in chemicals we cannot smell, in patterns we cannot see. Yet these signals structure the lives of insects as surely as language structures our own lives, creating social bonds, enabling cooperation, and allowing individuals to navigate their complex world. Social insects and colony life. Some insects have evolved beyond solitary existence to form societies of remarkable complexity and coordination. These social insects, primarily ants, bees, wasps, and termites, live in colonies that can number from a few dozen to many millions of individuals. Within these colonies, individuals are divided into castes that specialize in different tasks, creating a division of labor that allows the colony to function as a coordinated unit. A superorganism that can accomplish feats no individual could achieve alone. The hallmark of advanced social insects, called eusocial insects, is the presence of overlapping generations living together, cooperative care of young, and division of individuals into reproductive and non-reproductive castes. In a honeybee colony, for example, there is typically one queen who is the sole reproductive female. A few hundred to a few thousand males called drones whose only role is to mate with queens, and tens of thousands of workers, sterile females who perform all the tasks necessary to maintain the colony. The worker caste in bees and ants is further divided by age and by behavior into subgroups that perform different tasks. Young worker bees spend their first days of adult life cleaning cells in the comb, preparing them for the queen to lay eggs or for storage of pollen and honey. After a few days, glands in their head begin producing royal jelly and they become nurse bees feeding larvae. Later, they develop wax glands and become builder bees, constructing and repairing comb. Still later, they become guard bees, standing at the hive entrance and challenging incoming bees to verify they are nestmates. Finally, in the last weeks of their lives, they become forager bees, leaving the hive to collect nectar, pollen, water, and propolis. This age-related division of labor, called temporal polyethism, means that each worker bee performs a sequence of tasks throughout her life, with the tasks matched to her age and physiological state. The progression is not rigid, however. If the colony's needs change, workers can reverse course, with older bees reverting to nursing or younger bees precociously becoming foragers if those roles are needed. Ants show similar age-based task allocation, but they also have physical castes in many species. Some ant species produce workers of different sizes, with large-headed soldiers specialized for defense and small-headed workers specialized for brood care or foraging. These size differences are determined during larval development by the amount and type of food the larvae receive. A larva fed abundantly develops into a soldier. A larva fed less develops into a minor worker. The colony adjusts the ratio of soldiers to workers based on current needs, producing more soldiers when under attack and more workers when food is abundant. Termites are unique among social insects because both males and females are present in the worker caste, unlike ants, bees, and wasps where all workers are female. Termite colonies have a king and queen, both of which are long-lived reproductive individuals. The queen in some species becomes enormously enlarged, her abdomen swelling to many times the size of her head and thorax, turning her into an egg-laying machine that produces thousands of eggs per day. She is immobile and entirely dependent on workers to feed and care for her. The social organization of these colonies depends on chemical communication. The queen's pheromones maintain colony cohesion and regulate the development of workers, suppressing their reproductive potential. If the queen dies, her pheromone disappears and workers respond by raising new queens or, in some ant species, by activating their own ovaries and laying eggs. Trail pheromones organize foraging as workers mark paths to food sources and recruit nestmates to help exploit them. The cooperation within a social insect colony is made possible by genetic relatedness. In bees, ants, and wasps, which have a haplodiploid sex determination system, females develop from fertilized eggs and males from unfertilized eggs. This system creates unusual genetic relationships. Sisters share, on average, 75% of their genes, more than the 50% that mothers and daughters share. This high relatedness among sisters favors the evolution of worker behavior, because by helping to raise sisters, workers are propagating their genes more effectively than they could by producing their own offspring. This genetic explanation, developed by W.D. Hamilton and others, helps explain why eusociality has evolved multiple times in the Hymenoptera, the order that includes bees, ants, and wasps, all of which have haplodiploid sex determination. The high relatedness among sisters reduces the genetic cost of sterility and increases the benefit of helping relatives reproduce. The coordination of tasks within a colony emerges from relatively simple rules followed by individuals without any central control or planning. No ant knows the overall state of the colony or directs the activities of other ants. Instead, each worker responds to local cues, to the pheromones she encounters, to the interactions she has with other workers, and from these local interactions, colony-level patterns emerge. When a food source needs to be exploited, individual ants that find food lay trail pheromones. Other ants follow these trails, and if they also find food, reinforce them. The trail becomes a highway of workers moving food back to the nest. When the food is exhausted, ants stop reinforcing the trail and it fades away. This self-organized system is robust and flexible, adapting automatically to changing conditions. Task allocation similarly emerges from individual responses to stimuli. An ant is more likely to engage in a task if she frequently encounters the stimuli associated with that task. Ants that often encounter hungry larvae are more likely to feed larvae. Ants that often encounter garbage are more likely to remove garbage. The threshold at which an ant responds to a stimulus can vary among individuals, creating behavioral variation that ensures all necessary tasks are performed. The nest architecture of social insects is often complex and serves multiple functions. Ant nests can extend several meters underground, with chambers for brood, chambers for food storage, and tunnels for ventilation and access. Termite mounds in tropical regions can be meters tall, constructed from soil particles cemented together with saliva and feces. These mounds have sophisticated ventilation systems that maintain stable internal temperatures and gas concentrations, creating a suitable environment for the fungus that many termite species cultivate as food. Fungus farming ants in the genus Atta, the leafcutter ants, cut pieces of leaves and carry them back to their underground nests. They do not eat the leaves directly but use them as substrate to grow a fungus. The ants tend these fungus gardens carefully, weeding out unwanted fungi and bacteria, fertilizing the garden with their waste, and harvesting the fungus as food. This agriculture is complex and ancient, having evolved millions of years ago, and it allows leafcutter colonies to grow enormous, with several million workers in a single colony. Honeybee colonies build comb from beeswax, creating hexagonal cells with remarkable precision. The hexagonal shape is optimal for packing circles into a plane, and it uses the least wax for a given volume of storage. Bees construct these cells in darkness using only tactile cues, yet the cells are uniform and the comb is precisely vertical, allowing honey to be stored without dripping. Defense of the colony is a critical function in social insects, and many species have evolved specialized defensive strategies. Some ants have soldiers with large mandibles that can crush or sever the limbs of intruders. Others have soldiers that spray formic acid or other defensive chemicals. Honeybees defend their hives with stings, sacrificing their lives because the barbed sting remains in the victim, continuing to pump venom even after the bee has torn away and died. Termites have soldiers with bizarre modifications. Some have enormous jaws, others have nozzle-like heads from which they squirt defensive chemicals. In Nasuta terms, the soldier has a head shaped like a syringe, and it can spray sticky, irritating liquid at attackers, gumming up their antennae and mouthparts. These soldiers cannot feed themselves and must be fed by workers, but they provide essential defense for the colony. The reproductive strategies of social insects involve swarming or budding. In honeybees, when a colony grows large and strong, it prepares to reproduce by raising new queens. Before the new queens emerge, the old queen leaves the hive with about half the workers in a swarm, a dense cluster of bees that flies off to find a new nest site. Scouts search for suitable cavities, and the swarm makes a collective decision, choosing the best site from among those the scouts have found. Ant colonies reproduce by budding, with a new queen and a group of workers walking away from the parent colony to establish a new one, or by nuptial flights where virgin queens and males fly into the air to mate. After mating, the queen lands, sheds her wings, and begins searching for a suitable site to start a new colony. She digs a chamber, lays her first eggs, and raises the first workers herself, using energy reserves stored in her body. Once the first workers emerge, they take over foraging and brood care, and the queen dedicates herself entirely to egg-laying. The study of social insects has provided insights into cooperation, communication, and the emergence of complexity from simple rules. These insects demonstrate that sophisticated social organization does not require large brains or conscious planning. Through evolution and through simple behavioral algorithms, insects have created societies that rival our own in complexity and far exceed them in longevity. Some ant colonies live for decades. Some termite mounds are occupied continuously for centuries. These societies persist through generations of individuals, maintaining their structure and function across time. When we observe a line of ants marching across a sidewalk, we are seeing not just individual insects, but a piece of a larger organism, the colony. Each ant is both an individual and a cell in a superorganism. Her actions are her own, yet they serve the colony's needs. She will never reproduce, yet her genes continue in her sister's offspring. She has no understanding of the colony as a whole, yet her behavior contributes to its success. In the study of these insects, we confront questions about individuality, about purpose, about how complexity arises from simplicity, questions that resonate beyond biology into philosophy and into our understanding of ourselves. Defense mechanisms and survival. The world is dangerous for insects. They are hunted by birds, bats, lizards, frogs, other insects, spiders, and countless other predators. To survive, insects have evolved a stunning array of defensive strategies. Some hide through camouflage. Some advertise their toxicity with bright colors. Some mimic dangerous species. Some flee. Some fight. Some play dead. The diversity of insect defenses is a testament to the relentless pressure of predation and the creative power of natural selection. Camouflage, the art of blending into the background, is one of the most widespread defensive strategies. Many insects are colored and patterned to match their typical environment. Stick insects and walking sticks resemble twigs, complete with bark-like textures and even false leaf scars. They hold their legs and antennae in positions that enhance the illusion, remaining motionless for hours at a time. A bird searching for food might look directly at a stick insect and see only a twig. Leaf insects take camouflage even further. Their bodies are flattened and shaped like leaves with veins and sometimes even irregularities that mimic leaf damage or fungal spots. When they walk, they move slowly with a rocking motion that resembles a leaf swaying in a breeze. Some species can even change color over days or weeks to match the leaves they are eating, shifting from green to brown as leaves age. Cryptic coloration is not limited to mimicking plants. Moths often have wing patterns that resemble tree bark. When a moth lands on a tree trunk and folds its wings, it virtually disappears against the bark's texture. The peppered moth is a famous example, existing in light and dark forms. In industrial areas where tree trunks are darkened by pollution, dark moths are better camouflaged and more common. In clean areas, light moths predominate. This shift in population in response to changing environments is a well-documented example of natural selection in action. Some insects use disruptive coloration, patterns that break up the outline of the body and make it harder to recognize. Bold stripes, spots, or patches of contrasting colors can make it difficult for a predator to perceive the insect as a cohesive object. The eye is drawn to the high-contrast edges rather than to the overall shape, and the insect becomes harder to detect or to track. Countershading is another form of camouflage. Many caterpillars are darker on top and lighter below. When lit from above by the sun, the shadow on the underside is counterbalanced by the lighter color, flattening the appearance of three-dimensionality. The caterpillar looks less like a cylindrical object casting a shadow and more like a flat patch of color, making it less noticeable. Warning coloration, or aposematism, is the opposite strategy to camouflage. Insects that are toxic, distasteful, or dangerous often advertise this fact with bright colors and bold patterns. Monarch butterflies are orange and black, colors that warn birds of their toxicity. The caterpillars feed on milkweed plants, which contain cardiac glycosides, toxic compounds that the caterpillars sequester in their tissues. These toxins persist through metamorphosis into the adult butterfly. A bird that eats a monarch becomes ill, vomits, and learns to avoid orange and black butterflies in the future. Wasps and bees display warning coloration with their yellow and black stripes. These colors signal that the insect can sting, a painful experience most predators learn to avoid. The effectiveness of warning coloration depends on predators learning the association between color and unpleasantness, which requires that predators sample and survive the encounter. For this reason, warning-colored insects often occur in high densities, increasing the chance that a predator will encounter multiple individuals and learn the lesson. Mimicry occurs when one species evolves to resemble another, gaining protection by exploiting the predator's learned aversion. Bayesian mimicry involves a harmless species mimicking a dangerous or distasteful one. Many harmless flies mimic bees or wasps, with yellow and black stripes and buzzing flight patterns. Predators that have learned to avoid stinging insects also avoid these harmless mimics. The mimicry is often quite detailed, with the fly's antennae modified to resemble wasp antennae and the body shape sculpted to match. Mullerian mimicry occurs when two or more unpalatable species evolve to resemble each other. Several species of toxic butterflies might converge on the same warning pattern, sharing the cost of educating predators. A predator that learns to avoid one species automatically avoids all species with that pattern. This mutualistic mimicry benefits all participants by reducing the number of individuals that must be sampled for predators to learn avoidance. Some insects defend themselves with chemicals. Bombardier beetles produce a hot, noxious spray by mixing chemicals in a specialized chamber in their abdomen. The reaction produces heat and quinones, creating a boiling spray that the beetle can direct toward attackers with remarkable accuracy. The spray can deter even large predators and has been known to kill insects and small vertebrates. Ladybugs exude a foul-tasting fluid from their leg joints when threatened, a defensive behavior called reflex bleeding. The fluid contains alkaloids that are distasteful to predators. The bright red or orange coloration of ladybugs warns predators of this chemical defense, and most birds learn to avoid them. Stink bugs live up to their name by producing odorous compounds from glands on their thorax. When disturbed, they release these compounds, which smell unpleasant to us and to many predators. The smell can linger for hours, serving as a deterrent even after the bug has left. Behavioral defenses include fleeing, dropping, and thanatosis, or playing dead. When disturbed, many caterpillars drop from their plant on a silk thread, falling out of reach of predators. Weevils and other beetles tuck in their legs and drop to the ground when touched, relying on their hard exoskeleton to protect them from the fall and then remaining motionless, making them difficult to locate in the leaf litter. Click beetles have a unique escape mechanism. When on their back, they can snap a spine on their thorax into a groove, producing a clicking sound and launching themselves into the air. This sudden movement startles predators and often flips the beetle upright, allowing it to run away. Some caterpillars have intimidating displays. Certain swallowtail butterfly caterpillars have a forked organ called an osmeterium hidden behind their head. When threatened, they avert this organ, which resembles a snake's tongue, and it emits a foul smell. The sudden appearance of this bright, smelly structure often startles predators long enough for the caterpillar to be left alone. Eye spots, large circular markings that resemble vertebrate eyes, are found on the wings of many butterflies and moths. When the insect is resting, these eye spots are hidden. If a predator approaches, the insect suddenly opens its wings, revealing the eye spots. The predator, confronted with what appears to be the face of a much larger animal, often hesitates or flees. This moment of confusion allows the insect to escape. Some insects actively fight back. Driver ants and army ants swarm over animals that disturb their trails, biting with powerful mandibles. Hornets defend their nests aggressively, stinging repeatedly. The pain and injury inflicted by these attacks teaches predators to avoid these insects, and often the mere appearance or sound of these insects is enough to deter animals. Social insects gain defensive benefits from their numbers. A single bee might be easily eaten, but a hive of 50,000 bees that coordinate their defense through alarm pheromones is a formidable adversary. The collective defense of the colony protects the individuals within it, And the willingness of workers to die defending the hive makes evolutionary sense because they are protecting relatives who carry their genes. The evolutionary arms race between insects and their predators drives the diversity of defensive strategies. Predators evolve better detection, faster reflexes, immunity to toxins. Insects evolve better camouflage, more potent toxins, more convincing mimicry. This coevolution has been ongoing for hundreds of millions of years, and it shows no sign of ending. Each generation of predators and prey is tested, and those with even slight advantages survive to pass on their traits. When we encounter an insect, we are seeing the result of this long history of predation and defense. The green of a caterpillar is not random but precisely tuned to match the leaves it eats. The pattern on a moth's wing is a solution to the problem of avoiding detection. The bright colors of a beetle warn of chemical defenses accumulated over a lifetime of feeding on toxic plants. Every feature, every color, every behavior is part of a strategy for survival in a world filled with danger. The insects we see are the survivors, the descendants of countless generations that successfully avoided being eaten, and they carry within them the defensive innovations that made that survival possible. Insect roles in ecosystems. Insects are not merely inhabitants of ecosystems, but foundational elements that shape the structure and function of nearly every terrestrial and many freshwater environments. Their activities influence energy flow, nutrient cycling, plant reproduction, and the populations of countless other organisms. Without insects, ecosystems as we know them would collapse, and the world would be unrecognizable. Pollination is perhaps the most celebrated ecological service provided by insects. Approximately 3/4 of flowering plant species depend on animals for pollination, and the vast majority of these animal pollinators are insects. Bees are the most important pollination group, with over 20,000 species worldwide visiting flowers to collect nectar and pollen. As they move from flower to flower, they inadvertently transfer pollen, enabling fertilization and seed production. The relationship between flowers and their pollinators is often highly specific. Some flowers are shaped in ways that only certain insects can access. Orchids have evolved elaborate structures that ensure only particular species of bees or moths can pollinate them. These pollinators, in turn, have evolved specialized body shapes, tongue lengths, or behaviors that allow them to efficiently exploit these flowers. This coevolution has produced some of the most intricate biological partnerships on Earth. Moths are important pollinators of night-blooming flowers. These flowers often have white or pale colors visible in low light and produce strong fragrances to attract moths. Long-tongued moths hover in front of flowers, extending their proboscis deep into floral tubes to reach nectar, picking up pollen in the process. Some yucca moths have evolved an obligate mutualism with yucca plants, pollinating the flowers deliberately as part of their reproductive behavior while laying eggs in the flower's ovary. Flies, beetles, wasps, and even some ants contribute to pollination. Flies pollinate many early spring flowers and are the primary pollinators in Arctic and alpine environments where bees are scarce. Beetles pollinate ancient plant lineages like magnolias and water lilies. These less celebrated pollinators play essential roles in maintaining plant diversity and ecosystem function. The decline in pollinator populations in recent decades has raised alarm. Habitat loss, pesticide use, disease, and climate change all contribute to pollinator declines. The consequences extend beyond wildflowers to agricultural crops. Many fruits, vegetables, and nuts depend on insect pollination. The economic value of pollination services worldwide is estimated in the hundreds of billions of dollars annually. Protecting pollinators is not merely an environmental issue, but an agricultural and economic necessity. Decomposition and nutrient cycling are fundamental processes driven largely by insects. When plants and animals die, their tissues contain nutrients locked in organic compounds. Decomposers break down these compounds, releasing nutrients back into forms that plants can absorb. Insects are among the primary decomposers in terrestrial ecosystems. Dung beetles process animal feces, burying it in the soil where it enriches the earth and promotes plant growth. These beetles can bury thousands of kilograms of dung per hectare per year, removing it from the surface where it would otherwise harbor disease organisms and parasites. In Australia, where native dung beetles had evolved to process marsupial droppings, the introduction of cattle created a problem because the native beetles could not efficiently process cow dung. The solution was to introduce dung beetle species from other continents that were adapted to processing cattle dung. These introduced beetles dramatically improved pasture health and reduced fly populations. Carrion beetles, blowflies, and flesh flies process dead animals. Within hours of an animal's death, flies detect the odor and arrive to lay eggs. The larvae hatch and begin feeding on the decomposing tissues, consuming and breaking down the carcass. This activity accelerates decomposition, returning nutrients to the ecosystem that would otherwise be locked in dead tissue for extended periods. Wood-boring beetles and termites break down dead trees. Termites can digest cellulose, the main component of wood, with the help of symbiotic microorganisms in their gut. By tunneling through dead wood, they fragment it and expose it to further microbial decomposition. This process is essential in forests, where vast quantities of wood fall to the ground each year. Insects serve as food for countless other animals. Birds, bats, fish, amphibians, reptiles, and small mammals all depend on insects as a primary food source. Many bird species time their breeding to coincide with peaks in caterpillar abundance, feeding their chicks on the protein-rich larvae. Bats consume enormous quantities of flying insects each night, controlling insect populations and gaining the energy they need to survive. Aquatic insects form the base of freshwater food webs. Mayfly, stonefly, and caddisfly larvae graze on algae and detritus in streams. They are eaten by fish, which are in turn eaten by larger fish, birds, and mammals. The health of fish populations often depends on the abundance and diversity of aquatic insects. Anglers know this well, creating flies that mimic these insects to catch trout and other game fish. Some insects are predators or parasitoids that control populations of other insects. Ladybugs and their larvae eat aphids. Lacewing larvae feed on soft-bodied pests. Ground beetles consume caterpillars and slugs. These predators provide natural pest control, reducing the need for chemical pesticides and helping to maintain balance in ecosystems. Parasitoid wasps lay their eggs in or on other insects, and their larvae consume the host from the inside out. Each species of parasitoid typically specializes on a particular host or group of related hosts. By killing their hosts, parasitoids regulate herbivore populations and influence plant communities. The complexity of these interactions with multiple layers of predation and parasitism creates intricate food webs that stabilize ecosystems. Insects influence soil structure and health. Ants and termites excavate vast networks of tunnels, aerating the soil and improving water infiltration. Their nesting activities mix soil layers, bringing nutrients from depth to the surface. In some ecosystems, insects move more soil than earthworms, playing a dominant role in soil formation and maintenance. Seed dispersal by insects is less well known than by birds or mammals, but it is important for some plant species. Ants disperse seeds of many woodland wildflowers. These seeds have a fleshy appendage called an eleosome that attracts ants. The ants carry the seeds back to their nest, eat the eleosome, and discard the seed in their nutrient-rich refuse pile where it germinates. This mutualism benefits the plant through dispersal and nutrient provisioning and benefits the ant through food. Insects also influence ecosystems through herbivory. While we often view herbivorous insects as pests, they play natural roles in shaping plant communities. By consuming foliage, insects can reduce the competitive dominance of certain plants, allowing less competitive species to coexist. This herbivory increases plant diversity, which in turn supports greater diversity of other organisms. Outbreaks of herbivorous insects like bark beetles or gypsy moths can dramatically alter landscapes, killing large numbers of trees. While destructive from a human perspective, these outbreaks are natural disturbances that create opportunities for succession, for new growth, for species that colonize disturbed areas. In the long view of ecological time, these disturbances are part of the dynamics that maintain ecosystem diversity and resilience. The interconnectedness of insects with other species and with ecosystem processes means that changes in insect populations have cascading effects. Declines in pollinator abundance affect plant reproduction, which affects seed-eating animals, which affects predators. Loss of decomposer insects slows nutrient cycling, affecting plant growth. Changes in herbivore populations affect plant communities, which affects the entire food web. Insects are threads in the ecological fabric, and pulling those threads can unravel the whole. Understanding the roles insects play in ecosystems deepens our appreciation for these small creatures. They are not background noise in nature, but active participants shaping the world. Every flower we see may owe its existence to a pollinating bee. Every forest owes its nutrient cycling to beetles and termites. Every bird song may be fueled by the caterpillars and flies the bird has eaten. Insects mediate the relationships between sun and soil, between plants and animals, between life and death. And in doing so, they make the world habitable for all organisms, including ourselves. The diversity and distribution of insects. Insects represent more than half of all described living species on Earth, a level of diversity that defies easy comprehension. With approximately 1 million species formally described and perhaps several million more awaiting discovery. Insects outnumber all other animal groups combined. This extraordinary diversity is distributed across nearly every terrestrial habitat and many freshwater environments, from tropical rainforests to polar tundra, from deserts to mountaintops, from freshwater streams to temporary puddles. The orders of insects vary greatly in species numbers. The Coleoptera beetles are the most diverse, with over 350,000 described species. Beetles occupy an astonishing range of niches, including predators, herbivores, scavengers, and decomposers. Their success is often attributed to their hardened forewings, which protect the delicate hind wings and allow beetles to live in confined spaces like underbark, in soil, or in wood, environments that other insects cannot easily exploit. Lepidoptera, butterflies, and moths are the second most diverse order, with about 180,000 species. Their larval stages, caterpillars, are primarily plant feeders, and the diversity of Lepidoptera reflects the diversity of flowering plants. Nearly every plant species is fed upon by at least one moth or butterfly species, and the coevolution between these insects and plants has driven the diversification of both groups. Hymenoptera, which includes ants, bees, and wasps, contains over 150,000 species. This order is notable for including both solitary and highly social species. The evolution of eusociality has occurred multiple times within the Hymenoptera, and the ecological success of ants and social bees demonstrates the power of cooperative living. Diptera, flies, includes over 120,000 species. Flies are found in virtually every environment and have diversified into many ecological roles. Some are pollinators, some are predators, some are parasitoids, and some are decomposers. The larvae of different fly species feed on decaying organic matter, living plants, other insects, and even vertebrate tissue. This versatility has allowed flies to colonize habitats from Arctic tundra to tropical rainforests. Insects are found on every continent, including Antarctica, where wingless midges survive in mosses and under rocks. These Antarctic midges are the largest purely terrestrial animals on the continent, a reminder that insects can adapt to even the most extreme conditions. Tropical rainforests harbor the greatest diversity of insects. A single tree in the Amazon rainforest can host hundreds of insect species, and the forest as a whole may contain millions. The warm, wet conditions, the structural complexity of the forest, and the diversity of plants all contribute to insect diversity. Many rainforest insects are specialists, feeding on only one or a few plant species, and the high plant diversity supports correspondingly high insect diversity. Deserts, despite their harsh conditions, also support diverse insect communities. Desert insects have evolved remarkable adaptations to cope with extreme heat and scarcity of water. Some beetles collect water from fog by allowing it to condense on their bodies. Others burrow deep into sand during the day to avoid lethal temperatures, emerging at night to forage. Desert ants can navigate using polarized light and can withstand body temperatures that would kill most other animals. Aquatic insects are diverse in freshwater habitats but rare in the ocean. The reasons for this are not entirely clear, but they may relate to the physiological challenges of maintaining ion balance in seawater and to the dominance of crustaceans in marine environments. In freshwater, however, insects are abundant and diverse. Mayflies, dragonflies, caddisflies, stoneflies, and many beetles spend their larval stages in streams, rivers, lakes, and ponds. These aquatic larvae are indicators of water quality, with different species tolerating different levels of pollution. Islands often have unique insect faunas. Isolation allows populations to diverge, and over time, island insects can evolve into species found nowhere else. The Hawaiian Islands, for example, have hundreds of endemic insect species, including many flightless moths, beetles, and crickets. The evolution of flightlessness is common on islands, perhaps because strong winds can blow flying insects out to sea, selecting for individuals that stay on the ground. Caves harbor specialized insect communities adapted to perpetual darkness. Cave-dwelling insects, called troglobites, often lose their eyes and pigmentation, becoming pale and blind. They navigate by touch and chemical cues and feed on organic matter washed into the cave or on the guano of bats. These insects provide insights into evolution in extreme environments and into the sensory basis of behavior. Human-modified environments, including cities, agricultural fields, and buildings, have their own insect communities. Some insects thrive in these habitats, becoming pests or benefiting from resources humans provide. Cockroaches, ants, and flies are common in urban areas. Agricultural fields host both crop-feeding pests and beneficial insects like pollinators and predators. The insects that succeed in human-dominated landscapes are often generalists, capable of exploiting diverse or novel food sources and tolerating disturbance. The longevity of insects varies widely among species. Mayflies famously have very short adult lives, sometimes living only a few hours after emerging from their aquatic nymphal stage. In contrast, queen ants and termites can live for decades. The queen of some termite species may live for 50 years or more, continuously laying eggs and being tended by workers. This extreme longevity is rare in insects, but it demonstrates the range of life history strategies that evolution has produced. Cicadas exhibit some of the longest and most precisely timed life cycles. Periodical cicadas emerge in mass every 13 or 17 years, having spent more than a decade underground as nymphs feeding on tree roots. The timing is so precise that emergence years are predictable decades in advance. This long life cycle and synchronized emergence are thought to be adaptations to overwhelm predators and ensure that enough individuals survive to reproduce. Bioluminescence, the production of light by living organisms, is found in several insect groups. Fireflies are the most familiar bioluminescent insects, but there are also glowworms, certain click beetles, and some springtails that produce light. The light is produced by chemical reactions involving luciferin and luciferase, and it is used primarily for communication, especially for attracting mates. Different firefly species produce different colors of light, ranging from yellow-green to amber to orange. The color is determined by slight variations in the luciferin molecule or the luciferase enzyme. In some species, both males and females glow, while in others, only one sex produces light. The rhythmic flashing patterns are species-specific, and fireflies can recognize potential mates by the timing and color of flashes. Railroad worms, the larvae of certain click beetles, have a row of light organs along their sides that glow green and a red light on their head. This multicolored bioluminescence is unique among insects and gives these larvae an otherworldly appearance. The function of the lights is not entirely clear, but they may deter predators or help the larvae locate each other. The distribution of insects is influenced by climate, vegetation, geology, and history. Insects that cannot tolerate cold are restricted to warmer regions. Insects dependent on specific plants are found only where those plants grow. Mountain ranges, rivers, and deserts can act as barriers, isolating populations and leading to speciation. The current distribution of insects reflects both current environmental conditions and the history of continental drift, ice ages, and other large-scale events. Climate change is already altering insect distributions. Ranges are shifting poleward and upward in elevation as insects track suitable climates. Some species are expanding into areas where they were previously absent, sometimes with ecological consequences as they interact with species that did not co-evolve with them. Other species are declining or going locally extinct as their habitats become unsuitable. The diversity of insects is a testament to the versatility of the insect body plan and to the power of evolution to produce endless variations on a theme. From the bioluminescent glow of fireflies in the evening to the patient work of dung beetles in the soil, from the delicate flight of butterflies to the cooperative labor of ants, insects have explored nearly every possible way of making a living on land. They have adapted to deserts and rainforests, to caves and mountaintops, to freshwater and to the driest places on Earth. They have evolved to eat nearly every type of organic matter that exists, to live in association with nearly every type of organism. Their diversity is not merely a number, a count of species, but a reflection of life's endless capacity for innovation, adaptation, and persistence across hundreds of millions of years. When we observe an insect, we are seeing one small thread in an immense biological tapestry of diversity. That beetle crawling across the path belongs to one of hundreds of thousands of beetle species. That butterfly visiting a flower represents one expression of a lineage that has diversified into every color, every size, every pattern imaginable. Behind each individual insect lies a history of evolution, adaptation, and survival that connects it to ancestors stretching back to the Devonian period, to a time before flowers, before birds, before mammals, when insects were already ancient. The distribution and diversity of insects remind us that the world is fuller, richer, and more complex than we usually perceive. In every habitat, from the soil beneath our feet to the canopy far above, insects are present, living their lives, fulfilling their roles, continuing lineages that have persisted longer than most other forms of complex life on Earth. They are the small creatures that inherit the Earth, that have inherited it for hundreds of millions of years, and that will likely continue to inherit it long into the future. A gentle closing. As our journey through the world of insects comes to a quiet end, we find ourselves surrounded by a deeper understanding of these remarkable creatures. The mosquito that hums in the evening, the moth drawn to lamplight, the beetle walking across the forest floor. Each of these insects carries within itself hundreds of millions of years of evolutionary history. A body plan refined across geological ages, adaptations tested and proven through countless generations. We have explored how insects breathe without lungs, using networks of tubes that bring oxygen directly to their tissues. We have seen how they sleep and rest, guided by circadian clocks that tick away inside clusters of neurons in their tiny brains. We have discovered their senses, so different from our own, perceiving ultraviolet light and chemical signals we cannot detect, constructing a sensory world both alien and perfectly suited to their needs. We have learned about their exoskeletons, external frameworks that provide protection and structure, that must be shed and regrown as they develop. We have witnessed the extraordinary transformations of metamorphosis, where caterpillars dissolve and reform as butterflies, where grubs become beetles, where life cycles unfold in stages so distinct they seem like different organisms entirely. The communication systems of insects have revealed themselves to us. Languages of pheromones and sounds, of vibrations through plant stems and flashes of bioluminescent light. We have seen how some insects have transcended solitary life to form societies of breathtaking complexity. Colonies where millions of individuals cooperate as one, achieving together what no individual could accomplish alone. We have examined their defenses, the camouflage that makes them invisible, the warning colors that advertise toxicity, the chemical weapons they deploy against predators. We have recognized their essential roles in ecosystems, as pollinators ensuring the reproduction of plants, as decomposers returning nutrients to the soil, as food for countless other animals, as threads woven throughout the fabric of life on Earth. And we have contemplated their diversity, the million described species and the millions more that remain unknown, distributed across every terrestrial habitat, adapted to every conceivable way of life, from the frozen tundra to the scorching desert, from temporary rainwater pools to ancient forests. Insects are not merely small creatures that happen to share our world. They are fundamental components of the ecosystems that sustain all life. They pollinate the plants that produce much of our food. They break down organic matter and recycle nutrients. They provide food for birds, fish, and countless other animals. Without insects, the world as we know it could not exist. These small animals, often overlooked, sometimes feared or dismissed, are in fact marvels of evolution and adaptation. They have solved problems we struggle with, have developed technologies we try to emulate, have created societies we study to understand the principles of cooperation and organization. In their compound eyes, in their chemical communication systems, in their flight mechanisms, in their metamorphosis, we find inspiration for engineering, for computing, for understanding ourselves. As you drift towards sleep, you might imagine the world of insects continuing all around you. In the darkness, moths navigate by starlight. In the soil beneath you, beetle larvae feed and grow. In distant forests, ants tend their fungus gardens in underground chambers. On every continent, in every habitat, insects are living their quiet lives following patterns established millions of years ago, continuing the ancient rhythms of feeding and growing, of communicating and reproducing, of surviving and adapting. The cricket's chirp that marks the evening temperature, the butterfly's wing that catches the morning sun, the ant's patient carrying of a seed. Each of these moments is part of a larger story, a story that began before the dinosaurs and that continues into an unknowable future. Insects are the small witnesses to Earth's history, the small inheritors of time, and in understanding them we understand something essential about life itself, about persistence, about adaptation, about the endless creativity of evolution. Let this knowledge settle gently into your mind as you rest. There is no need to remember every detail, every fact, every name. Simply allow yourself to appreciate that the world is inhabited by these remarkable creatures, that they have been here far longer than we have, that they will likely persist long after we are gone, and that their quiet presence enriches every ecosystem, every habitat, every moment of life on this planet. If you enjoyed this exploration of the insect world, if you found peace in these facts delivered softly into the night, consider subscribing to Nighttime Science. We journey together through the quiet corners of scientific knowledge, exploring topics that calm the mind while expanding understanding. A gentle like on this video helps others find this content when they need relaxation and learning combined. Thank you for spending this time in the world of insects. May your rest be peaceful, your dreams be calm, and your appreciation for the small creatures that share our world be deepened. Good night.